Hello, I'm Professor Matthew Schmidt, and this is Genetics. And in today's session, we're going to revisit Mendel. And what I mean by that is we're going to take a look at altered dominance relationships and multiple alleles, two situations where Mendel's principles, at least initially, seemed like they may be in question, but eventually it was understood how we can interpret some of these strange things in terms of what Mendel said. So you'll see more what I mean as we go along with this. So exceptions and extensions. Often textbooks have chapters named this, exceptions and extensions. I put exceptions in quotation marks because it turns out really there are no true 100% real exceptions to Mendel's laws. That's what happens. But whenever you see something strange or not exactly what you would have predicted, it seems like an exception. An extension just means taking it a little further than Mendel might have taken it. So as you can see here, as experimentation continued, Mendel's laws did seem to hold true for all sexually reproducing eukaryotes, but as they used different organisms and did different types of experiments, seeming exceptions were discovered. So when they saw these exceptions, the big question is this one right here. Do these exceptions or seeming exceptions invalidate the laws, meaning that Mendel's principles only apply, in certain cases and sometimes they don't? Or can we interpret these apparent aberrations in terms of Mendelian principles? For example, we've already seen that with sex linkage, that was an apparent exception because Mendel never said anything about only females expressing one trait or the ratios being different between males and females, etc. But we realized that in that case, the reason we were seeing things that Mendel had never said was because the genes were located on the X chromosome, and we were dealing with animals, in this case, that were either male or female. So it wasn't that Mendel's laws were invalid all of a sudden. It was just that we had to understand them in a new context. So his laws remain unaltered. We just said, wow, there's sex linkage. That's an extension. What seemed to be an exception became an extension of Mendel's laws. So we're going to see a couple of other things in this session that were noted and are often the types of things that are asked on exams. So let's go right into them. One of the most important things that Mendel couldn't have understood, this is the phenomenon of altered dominance relationships. So I think you'll agree. So far, and as far as Mendel said, we've considered a situation where there were always two alleles and one was dominant to the other, right? For all of Mendel's traits, all 7 of them, that's the way it was. 2 different alleles, one always completely dominant to the other. It turns out that this is not always the case. So let me give you an example and we'll see how this might impact the results of a cross. In snapdragons, which are a type of flower, pure breeding red flowered plants, so in this case snapdragons happen to have red flowers and they happen to have white flowers. So if you took a pure breeding red flowered plant and you had a pure breeding white flowered plant line, you can have this just like Mendel had his purple flowered pea plants and white flowered pea plants. If this went along exactly as Mendel said, if you crossed the red pure breeding line with the white, you might expect the F1 all to look red, right? Because it resembled one of the parents, and while it could be either one, Mendel, the offspring looked like the purple parent in the F1. But in the case of snapdragons, what we see is that the F1 all are pink. And here you see a pretty pink snapdragon flower. Now, it's sort of a good thing that Mendel didn't use snapdragons, or else there may have been some problems, because this almost looks like that initial idea of blending inheritance that we said was so prevalent before Mendel did his work, right? That you sort of mix everything up and put them together, and then the offspring are somewhat in the middle. It does look like that. It's not really what's going on. What's going on is this phenomenon known as incomplete dominance. And what this means is that neither of the alleles really are completely dominant to the other. They're both sort of equal strength, for lack of a better term. So I'm going to tell you, I'll give you an explanation of how this works at the molecular level when we're done with this part. But for now, let's just see how it affects the results of a cross. So as I just said, incomplete dominance occurs when one allele is not completely dominant. That's what Mendel, I don't know if he used the word complete dominance, but that's what it means. In this case, both are equal contributors of equal strength, and the phenotype observed in a heterozygote is somewhere intermediate between that of the homozygous parents. It does resemble blending inheritance. The reason it's not is because, as we're going to see, the alleles are not diluted in any way. It's just that something a little bit different is going on here. What is going on? Let's find out. So diagrammatically, just because we had these colors, say this is just meant so you understand the concept. A parent that's dark blue in this case mates with a parent that's white. In this case, you do get offspring that are somewhere in between, like a light blue type of a thing. All right, even though in snapdragons they aren't blue necessarily, but you get that idea, right? Let's see how it actually happens that way. So here's a picture that shows actual snapdragon flowers. We couldn't find one that showed red, white, and pink, but this is just to give you an idea that there is phenotypic variation in what a snapdragon flower actually looks like, because the ones we have here don't look exactly like that. Antirrhinum majus, the snapdragon. Pretty. Okay, so let's look at a cross in the exact same format as Mendel would have done it. And what I mean by that is, this doesn't look red, I'll grant you, but it's supposed to be a red flowered plant, and you can tell the difference between it and a pink one, and we'll see in a minute. We're doing this under the assumption that things are working in a perfectly Mendelian way. So that's why I randomly chose the letter A and said, look, if we would guess based on what Mendel said, we would say that the red flowered snapdragons are big A, the whites are little a, right? And we know for a fact that in the F1, this parent over here can only give big A, this parent can only give little a. So all of the F1 are going to be heterozygotes, right? Now, here's where the interesting part comes in. If we were doing it and the results went according to what Mendel said, we would probably expect all of the F1 offspring to resemble one of the parents, that which was dominant, which we were guessing was red. But in fact, they don't look that way. They both look pink. I apologize if the colors aren't coming through exactly as they should. But that's the deal, that they look pink, and then we have to follow it through, because remember, Mendel then took the F1 and crossed them with themselves or with someone from the same generation, the brothers or sisters. So both parents can put either a big A or a little a into the gamete as we do the Punnett square. And if we fill in the boxes, we'll find out, and it's not very shocking, that we're going to get 1/4 that are big A, 2/4 or 1/2 that are big A, little a, and 1/4 that are little a. Genotypically, this is exactly what we saw in all of the crosses that Mendel did, and this is why we're saying that this is not, in fact, an exception to Mendel's law. It's an extension. It's an extension, because really the only thing different here is, look at the phenotypes that we get here. We get 1 red, 2 pinks, and 1 white. If this were a normal Mendelian complete dominance situation, is it not true that all 3 of these, because they had at least 1 big A, they would look the same? That's how we got the 3-to-1 ratio. The only thing that's happening here different, and I'm not saying it's, it's phenotypical.